If you've been reading MS research news lately, you might have come across the word remyelination. Now, most of us with MS have heard of demyelination, but perhaps not remyelination. I'm here today with Dr. Nick Cunniffe, who's been involved in some of the remyelination research recently. Hi, Nick. Thanks for talking to us today.
Hello. Good afternoon.
Could you tell us, what is remyelination?
Absolutely. So I think perhaps the best starting point is to say that MS is primarily a disorder where the myelin lining of nerve cells becomes damaged, and we know that as demyelination, exactly as you said. Now, regeneration or repair of myelin can occur, and we actually will have stem cells in our brains and spinal cords that are able to migrate to areas of damage and start this repair process. But unfortunately, this remyelination process, this repair, fails in the majority of people living with MS. So remyelination or a remyelination treatment is important because it's a therapy that might actually enhance the repair process and allow recovery of some function.
So if there was a drug that would remyelinate, what would we expect it to do for people with MS?
So the rationale for remyelinating therapies are twofold. And I think this relates back to the two main functions of myelin and also the cells that make the myelin. We know these as oligodendrocytes. So the first function of the myelin lining of nerves is that it allows information to be sent along nerve fibers over distance in a reliable fashion. But the second and often underappreciated role of myelin is that it actually insulates and protects the underlying nerve fiber itself. And it's the demyelinated area of nerve that leads to vulnerability of that nerve and can lead to degeneration. So a remyelinating therapy, we believe, will have two key effects. First, that it will allow nerves that have been damaged by MS to recover function, and that will hopefully reduce MS symptoms. But also, by remyelinating nerve fibers, it will protect them from degenerating and therefore reduce progressive disability in MS.
So if somebody already had progressive MS, would remyelinating therapies actually help them as well?
Yeah, we believe so. We do believe so. But there is a caveat, I would say, to the question of how remyelinating therapies will affect progressive MS. And that's to say that in order for remyelination to occur, you do need to have an intact nerve cell. And that's important because this oligodendrocyte cell has to come and recognize the nerve cell and appropriately put the myelin around that cell. Now, in progressive MS, some nerve fibers have already degenerated, and that's what's given rise to the progressive disability, but not all. So I think it certainly will be effective in people with progressive MS, but we do not know how effective compared to people perhaps treated earlier on in the illness.
Right. Another word that we can hear sometimes in this is neuroprotection. What's the difference between remyelination and neuroprotection?
Yeah, I mean, neuroprotection really is just an umbrella term for a treatment that protects nerve cells from degenerating. So there are several possible ways of achieving neuroprotection, protecting the nerves, but one of these is remyelination.
So recently we've heard some news about this trial that's been going up in Cambridge, which was the Bexarotene trial. Could you tell us a brief outcome of that trial?
Yes. So the Bexarotene trial, otherwise people might have seen it described as the Cambridge Centre for Myelin Repair Trial number 1, so abbreviated to CCMR1. It's one of the really exciting recent results we've had in the field. So its story in Cambridge actually goes back over a decade when one of the laboratories there showed that a particular target on the cells that are able to repair damage We know this is RXR gamma. We know that if you target that particular part of the cell, it can enhance remyelination in animals. Now, there's no drug that targets that precise target, but there is a drug called Bexarotene that is a treatment for a rare type of skin cancer that targets the RXR gamma, but among a few other related targets. And that was the drug that we tested in the CCMR1 trial, really to determine whether the remyelination that was seen in animals also occurs in people with MS. So this trial occurred in 2 centres, both Cambridge and Edinburgh, and involved 52 people with MS. And half of those received Bexarotene, half of those received placebo. And we tested whether the drug promoted remyelination in 2 ways. We looked at the structure of nerve fibres with an MRI scan. And second, we can look at the function of nerve fibres, which improves with remyelination by using a special test of the eyes known as the visual evoked potentials. And our results showed that Bexarotene did lead to remyelination, but that this depended on how damaged the nerves were and also where the damage occurred, where the damage was within the brain. The remyelination appeared to be greatest in the grey matter regions of the brain compared to the white matter regions of the brain. The disappointing side of the trial was that the drug itself caused several side effects. It caused everyone that received the drug to develop underactive thyroids that did recover after stopping the drug, but it was significant. People did experience, for example, fatigue and reduced energy levels as a result of that. And also it led to elevated levels of the fats in the blood, the triglycerides. And while these are recognized side effects of Bexarotene, they occurred slightly more frequently in our trial compared to the people who had received the drug for cancers. So I think our conclusion was that this is a really important target to promote remyelination, but that we do first need to develop a drug capable of doing that without the side effects that we came across.
But still, exciting outcome to see that it sort of worked. So, what other trials are happening in this field then?
Yeah, so to pick up on that point, I mean, there have been a few others, and the Bexarotene trial, CCMR1, has been the first to show evidence of remyelination with converging evidence, so evidence from both the brain scans and the VEPs. Both showed a positive effect, and that hasn't been done before. But there are others. I think perhaps the most noteworthy trial was the Rebuild study, which used an antihistamine known as clemastine, and it showed a small beneficial effect on the VEP, the visual evoked potential that I mentioned. There are several other trials being undertaken at the moment. There's another trial using clemastine, testing it in people who've had recent episodes of optic neuritis. There have been trials using a drug known as opicinumab, otherwise known as an anti-LINGO-1 antibody. There have been several trials of that, and there's another one ongoing at the moment, tinkering with doses to see what might help people with MS. There's another trial in America and Canada, I believe, using nanocrystalline gold, which is a very interesting, intriguing way of promoting remyelination. So I think, yeah, lots of exciting research going on. We were looking forward to seeing the results from other groups elsewhere.
So clearly a happening field, which is very exciting. So another trial that we've heard about that you guys are involved in is another repurposed drug, Metformin. Can you tell us about that trial?
Yes, absolutely. So that trial is going to be called CCMR2. Not very inventive, but that's the name of the trial that we've gone for. Again, the rationale for it comes from the same laboratory that showed that Bexarotene worked in animals first. And they've shown that Metformin, an anti-diabetes drug, can promote remyelination in animals in a similar way. It works in a slightly different way compared to Bexarotene. Actually, what it does is it tells the stem cells that we have in our brains to be in a position to respond to the signals or instructions that can tell them to start with the repair process. So it makes the stem cells receptive to the instructions to commence repair. So in actual fact, we think this drug is best given as a combination treatment. With metformin, but also alongside clemastine, which is the drug that I mentioned before. So the clemastine acts as a signal to tell the stem cell to commence repair, but the metformin allows that stem cell to be in a position to respond to the clemastine. So it's almost a synergistic effect of the two. I think what's exciting is that metformin is an anti-diabetes drug that's widely prescribed. We know it's safe. It's generally speaking very tolerable, including in people living with MS that also happen to have diabetes. And it's exciting to see it put into a trial to see if it enhances remyelination in people with MS as well as it did in animals. I mean, the plan for the trial is similar to the last study that we've done. It's going to involve 50 participants again, but this time it's just going to be based in Cambridge. Everyone will have relapsing remitting MS, but everyone must be on a disease modifying treatment to be included or to be eligible. The reason being is we want to make sure that the inflammation of MS is controlled so we can study the repair process happening on account of the metformin and clemastine in isolation. And again, we're going to be using both the brain scans and the eye tests, the visual evoked potentials, to test if the drug's having a beneficial effect compared to those on placebo.
Do you think if trials like this are successful for relapsing remitting MS, that there would be trials for people with progressive MS as well?
Yeah, absolutely. So this is an important question. So why is the trial happening? Why is the trial focusing first on people with relapsing remitting MS? And of course, treatments for progressive MS is the greatest unmet need that we have in the MS community at the moment. I think the answer to that goes back to a point I made before about the proportion of nerve fibers that have degenerated. So in relapsing MS, more nerve fibers will be in a position that are able to be remyelinated. So we should be able to measure a biological effect in a clinical trial with a smaller number of participants. That's not to say that the drug's not going to work in progressive MS. It just means that it would be harder to demonstrate in a clinical trial with a small number of participants. And that's because in order to show that this drug works, we're using combinations of MRI scans and visual evoked potentials rather than seeing if it's improving, for example, disability. That would be for a phase 3 trial rather than the phase 2 trials that I'm mentioning here. So absolutely, it all comes back to what the objective of the clinical trial is. So this trial, and most of these trials, really are seeking to demonstrate, do these drugs have a biological effect in humans? We know they work in animals. Do they have the same effect in humans? If the answer to that is yes, then they need to be tested in large clinical trials, which will undoubtedly include people with progressive MS. But for this first comparatively small trial, we think it's best to first do that in a small number of people with relapsing MS.
And so you mentioned all the different phases and things like that. If trials are successful, how long do you think it will be until we actually see any drugs sort of out on the market that will promote remyelination?
Yeah, the question of how long is hard to say. I think the benefit of using the word repurposed, but drugs that like metformin, the diabetes drug, clemastine, the antihistamine, is that they're already licensed. We know they're safe in people. So if they're shown to be effective in clinical trials, then there aren't perhaps as many barriers to their being introduced to the MS community as, for example, a novel drug, a drug that's not been tested before in people before. But unfortunately, the trials that I'm mentioning, they do take some time. For example, I don't anticipate that the CCMR2 trial will have results for a couple of years. Unfortunately, the COVID pandemic has delayed things significantly for us, but we hope to be starting in the first quarter of 2021. And also there are some unanswered questions for us as well. We don't know, as I mentioned, exactly who's going to benefit most from these drugs. Hopefully everyone, but we don't know that. We don't know when they should be given. Should they be given continuously or just in pulses or just after relapse? We don't have answers to that, but people are working on those answers. But I think kind of reflecting a positive note at the end, rather than just citing all the challenges, I think, despite those challenges, it's now looking increasingly likely that a remyelinating treatment for MS is a matter of when rather than if. So it is an exciting time for the MS community, I feel.